Hello friends, today we will be discussing the cell structure and function. So when we discuss about the cell structure and function, we should know that there are two different kinds of cell. One is prokaryotic cell and other is the eukaryotic cell. Prokaryotic cells are the primitive cells, whereas eukaryotic cells are basically the multicellular. So when we talk about prokaryotic cells, they are usually unicellular. Okay, they are unicellular, and eukaryotic cells are majorly multicellular. So in prokaryotic cells, they do not have a cell membrane, a specific not a cell membrane, the nuclear membrane. They do not have a particular nuclear membrane or a well-defined nucleus. So they have a nucleoid in the place of it, and there is circular DNA which is present in the form of a plasmid. Okay. Then after that, in prokaryotic cells, nucleus is absent, whereas nucleus is present in eukaryotic cell. And in prokaryotic cell, the cell organelles are also absent, like plasmid, mitochondria, and etc. That is also not there. Whereas in eukaryotics, all of them are present. All the organelles are present. Okay. So coming to eukaryotic cell, when we talk about eukaryotic cell, this is a prokaryotic cell. So just to make a change, this is not eukaryotic cell, this is a prokaryotic cell. Okay, so this is a prokaryotic cell. So in a prokaryotic cell, the cell membrane is responsible for regulating materials move into and out of the cell. There is a nucleoid, the DNA with ends that come together to form a circle, and it is not wrapped around proteins, that is naked DNA. And cytoplasm is a gel-like fluid substance, the site of metabolic reactions. Then we have ribosomes, which build proteins during translation. Then we have plasmids, which is an extra piece of circular DNA that can be shared between the bacteria, often that contain genes for antibiotic resistance. We have the cell wall, which provides shape and allows the cell to withstand turgor pressure without bursting. We have pili that attaches to the surface, swaps DNA with other cells, and may be used to harpoon DNA into the environment. Then we have the capsule, which helps the cell keep from dehydrating and adhere to surfaces. Then we have the flagella for long extensions used in cell locomotion. So these are the characteristics of a prokaryotic cell. The next we have the eukaryotic cells. So eukaryotic cells are of two types, plant cells and animal cells. So plant cells are found basically in the Monera, bacteria, blue-green algae, mycoplasma, and all those, whereas an animal cell you can find in protista, fungi, plantae, and animalia. Okay, now nucleus is available. Nucleus is like a nucleoid is present, no nuclear membrane is there in plant cell, whereas in animal cell it is available. Chromosome, single chromosome in plant cell, whereas more than one chromosome is present in the animal cell. Okay, so these are the various differences that we have. Now coming to the plasma membrane. So we are talking about the structure and the functions of different organelles which are there. In the cell boundaries, we have plasma membrane or the cell membrane, which surrounds the cell and allows specific things to in and out. So it is selectively permeable. It's basically made up of phospholipids. It's there both in prokaryotics and eukaryotics. Cell wall is present only in the plants of the eukaryotes, and prokaryotes is present. It is made up of cellulose, and it gives extra support and protection to the cell. We have a nucleus. Nucleus is present in eukaryotes, both in plants as well as animals. And here the nucleus, it is the command center or the brain of the cell, which controls all the activities, and it contains DNA which gives instruction for making proteins. Then we have chromatins. Chromatins, you have a nuclear pore, and there is a chromatin fiber inside the nucleolus, and this chromatin fiber contains a genetic material, that is DNA, okay, which gets condensed to form the chromosomes. We have nucleolus. Nucleolus, it makes ribosomes, which is found inside the nucleus and found in eukaryotes. Then we have ribosomes that makes proteins. It's made up of RNA and can be free in cytoplasm or attached to endoplasmic reticulum, found both in eukaryotes and prokaryotes. Then we have the nuclear membrane. So it controls what enters and exits the nucleus. It surrounds the nucleus and found in eukaryotes. We have the centrioles, which is involved in cell protection. It's made up of microtubules. Two are paired at right angles to each other and found in eukaryotes, animal cell. Cytoplasm is a jelly-like substance. It's a site of chemical reactions that takes place and suspends all the organelles. It contains clear gel-like fluid called as cytosol. It's found both in eukaryotes and prokaryotes. Rough endoplasmic reticulum or round endoplasmic reticulum, it produces proteins and helps move molecules throughout the cell, found basically in the eukaryotes. Smooth endoplasmic reticulum produces lipids. It detoxifies proteins and helps to move the molecule throughout the cell, found basically in the eukaryotes. We have Golgi Apparatus, which is again found only in the eukaryotes. It works closely with endoplasmic reticulum. It's a flattened sac membrane, and the function is basically to process, packs and secretes proteins and lipids. Then we have vacuoles, which is empty space which stores food, water, waste, proteins, carbohydrates, etc. It helps to maintain homeostasis, and it is in eukaryotes where you have plants have a larger vacuole, animals have a very small vacuole. Lysosomes are suicidal sacs. It is used to digest old organelles and foreign objects, which is found in eukaryotes. We have a chloroplast, which is basically found in plants, which is used for the photosynthesis, and glucose is made over here. Okay, mitochondria, which is the powerhouse of the cell, found in eukaryotes. It breaks down ATP during cellular respiration. We have a cytoskeleton, which maintains the shape and supports the cell. It is made of microfilaments and microtubules found throughout the cytoplasm. It's found both in eukaryotes and prokaryotes. We have a cilia, which helps in the movement of cells or fluids. We have short hair-like fibers. Eukaryotes found in eukaryotes as well as prokaryotes. Even your bronchus, they have this microtubules which exhale out any debris that is there or removes any microbes when you cough. Then we have flagella, which is help for the locomotion or movement of the cell, which is a long hair-like fiber. So only animal cell which has this flagella is your sperm. Okay, so that's all the organelles that we have discussed today. Thank you. Just to go through your textbook, we have of class 9. So what is the cell made up of, and what is the structural organization of a cell? So if we are studying the cell under microscope, we will find various organelles of a cell. Like we have got plasma membrane, nucleus, cytoplasm. When we saw the plant cell, we saw this. Then we have plasma membrane or cell membrane. Plasma membrane or cell membrane. So this is the outermost covering of the cell that separates the contents of the cell from the external environment. The plasma membrane allows the permit of entry and exit of some materials in and out of the cell. It prevents the movement of some other materials. The cell membrane, therefore, is called selectively permeable. However, does the movement of substance take place into the cell? How do the substance move? Some substances like carbohydrates or oxygen, they will move across the cell membrane via the process of diffusion. Okay. And in the case of diffusion, we saw that there is a spontaneous movement of substances from a region of high concentration to a region of a low concentration. So something similar happens in the cells. For example, some substances like carbon dioxide, which accumulates in higher condition inside the cell. In cells external environment, the concentration of carbon dioxide is low as compared to the inside. So as soon there is a difference, what will happen? So the carbon dioxide inside and outside of the cell, so the carbon dioxide will move out of the cell from a region of a high concentration to a region of a low concentration. Also the oxygen also, it forms the same way. Water also obeys the rule of diffusion. So we have this membrane which is selectively permeable. Okay, and when water moves across, then it is known as osmosis. The movement of water across the plasma membrane is also affected by the amount of substance dissolved in water. So we have got three variations. One is isotonic solution, one is hypotonic and hypertonic. So this is this is a water body. This is a semi-permeable membrane. So if I have same amount of solutes on either side of the membrane, then I will say it is isotonic. But if the concentration on the side is lower, inside is lower, say this is inside and this is outside. Okay, so if inside is lower, I say it is hypotonic, and if it is higher, I say it as hypertonic. Okay, so here you see, so in the medium surrounding the cell, it has higher water concentration than the cell, meaning that the outside solution is very dilute. The cell will gain water by osmosis. Okay, such solution is hypotonic. So water molecules are free to pass across the cell membrane in both the directions. The medium has exactly same water concentration on either side, so there is no net movement. We say that is isotonic. And if the medium has lower concentration of water, then there is the water moving in, so that a very concentrated solution, the cell will lose water by osmosis. So this is hypertonic. So there is a loss of water. So in hypertonic, you see, so in hypertonic, when you see the water, the cells, so but more water will come into the cells. So water is coming into the cells. So it is hypertonic. If there is no net movement, it is isotonic. And if the movement is going out, so outward movement is, then the outward movement is hypertonic. Okay, so if it's moving out, it is hypertonic. Again, water crosses the cell membrane in both directions. So therefore what happens, in the hypertonic condition the cell will shrink, hypotonic condition the cell will enlarge itself. Okay, so that is that. So what are the activities that is seen? Osmosis with an egg. Remove the shell of an egg by dissolving it in dilute hydrochloric acid, and then the shell is mostly calcium carbonate with thin outer skin. Now encloses the egg. Put the egg in pure water and observe for 5 minutes. What do you observe? The egg swell. Similarly, now in this case, if you cannot do with an egg, I will tell you to do with a slice of potato. Take some few slices of potato, put it in water. So once you put it in water, leave it for a few minutes and you will see the potato slices have swelled up. Okay. Then put some salt into it, and you will see water is coming out. It is releasing water. You just mix the potato for some time, potato slices with salt and keep. You will see there is a release of water. So that is, the water is moving out whatever extra water is there. Okay. Now, unicellular freshwater organisms and most plants tend to gain water through osmosis. Absorption of water by plant roots is also an example of osmosis. Thus, diffusion is important in exchange of gases and water in life of a cell. In addition to this, cells also obtain nutrition from its environment. Different molecules move in and out of the cell through a type of transport requiring use of energy. Okay, so the plasma membrane is flexible and is made up of organic molecules called lipids and proteins. However, we can observe the structure of plasma membrane only through a microscope, electronic microscope. Okay, so now when there is a flexibility of cell membrane, it enables you to gulf in food and other materials from the external environment. It is known as endocytosis. You are taking in food, that is endocytosis, and when you are exhaling out, then it is exocytosis. Okay, next coming to the cell wall. So in plants, we basically have the cell wall, which is rigid outer covering, and this cell wall lies outside the plasma membrane. The plant cell wall is mainly composed of cellulose, which is a complex substance, provides structural strength to the plants. When a living plant cell loses water through osmosis, there is shrinkage or contraction of the contents of the cell away from the cell wall. So this phenomena is known as plasmolysis. So when there is a shrinkage of the cell, it's plasmolysis in plants. Okay. Then we have the cell wall, which permits the cells of plants, fungi and bacteria to withstand very dilute, that is hypotonic external media without bursting. In such media, the cell tends to take up water by osmosis. Now the cells will swell and they will build pressure against the cell wall. These walls exert equal pressure against swollen cell because of their walls. Such cells can withstand much greater changes in the surrounding than animals. Okay, now coming to the nucleus. Now so there is a nucleus. So in the chemical composition of the different regions of the cells, it gets colored differently. Some regions appear darker. Apart from iodine solution, we also use safranin, etc. So that area which is dark, that is known as, that you have seen is nucleus. When you saw the onion cell, so you will have the dark portion over here. So this is the nucleus. Okay, so the nucleus has double layered covering called nuclear membrane. The nuclear membrane has pores which allow the transfer of materials from inside of the nucleus to outside, that is cytoplasm. The nucleus contains chromosomes which are visible as rod-shaped structures only when the cell is about to die. Chromosomes contain information for inheritance of characteristics for parents to next generation. So chromosomes are hereditary materials. So chromosomes are composed of DNA and proteins. DNA molecules contain information which is necessary for constructing and organizing cell. The functional segment of DNA is called the genes. In a cell which is not dividing, the DNA is present as a chromatin material. Chromatin material is visible at entangled mass of threads, like a very confused mass of threads. So the nucleus plays a central role in the cellular reproduction. So here what happens, the process by which single cell forms two cells, so that is cellular reproduction that is occurring. Okay, so it also exhibits maturity, develops, and by directing chemical activities of the cell. Some organisms like bacteria, the nuclear region of the cell, it is very poor because we know there is no defined nuclear membrane that is there. There is only a nucleoid. Okay, so that is the prokaryote. Prokaryotic cells, they lack most of the cytoplasmic organelles. Fine, so that we have seen it. So the chlorophyll is photosynthetic prokaryotic bacteria is associated with the membranous vesicle but not with the plastids as in eukaryotes. Okay, then we have the cytoplasm. What is cytoplasm? It is a jelly-like substance which is there, wherein the chemical reaction takes place, and where it holds all the organelles. So each of the organelles, they perform specific functions. So they are enclosed by the membranes. All the organelles are enclosed by the membranes. In prokaryotes, the defined nuclear region, there is absence of defined nuclear region. So the membrane-bound cell organelles are also absent. So on the other hand, eukaryotic cell, the nuclear membrane as well as all the organelles are present. Okay, so then we have the cell organelles that are there. We are talking about what are the different cell organelles that we have. Every cell has membrane around it to keep its own content separate from the external environment. Large and complex cell, including cells from multicellular organisms, need a lot of chemical activities to support their complicated structure and function. To keep these activities of different kinds separate from each other, the cells use membrane-bound little structure within themselves. This is one of the feature of the eukaryotic cell that distinguish it from the prokaryotic cell, because prokaryotic cell do not have a nuclear bound organelles. So what are the different organelles we have? Endoplasmic reticulum, Golgi apparatus, lysosomes, mitochondria and plastids. Okay. Now coming to the first, endoplasmic reticulum. Endoplasmic reticulum is a large network of membrane-bound tubes and sheets. So it just looks like long tubes or oblong bags. The endoplasmic reticulum membrane is similar structure to the plasma membrane. So there are two types of endoplasmic reticulum, smooth and the rough endoplasmic reticulum. So the rough endoplasmic reticulum has ribosomes attached to the surface. Okay, the ribosomes which are present in all active cells are the sites of protein manufacture. So you have the endoplasmic reticulum. Okay, so you have the ribosomes attached on the surface of the rough endoplasmic reticulum, where there is the protein formation takes place. Okay, and smooth endoplasmic reticulum helps in the manufacture of fat molecules or lipids, which is important for the cell function. Okay, so now the proteins and lipids help in building the cell membrane. This process is known as membrane biogenesis. How the membrane is formed. Okay, the formation or genesis of biological genesis of membrane, that is membrane biogenesis, is that from the smooth endoplasmic reticulum there is the formation of cell membrane. Okay, so some other proteins and lipids function as enzymes and hormones. Although endoplasmic reticulum varies greatly in appearance in different cells, it always forms the network system. Thus, one function of endoplasmic reticulum is to serve as channel for the transport of materials between various regions of cytoplasm or between the cytoplasm and nucleus. So, the endoplasmic reticulum also functions as a cytoplasmic framework, providing the surface for some of the biochemical activities of the cell. In the liver, cells of groups of animals called vertebrates, smooth endoplasmic reticulum plays a crucial role in detoxifying many poisons and drugs. Okay, now Golgi apparatus. We have another which is known as Golgi apparatus. It is named after its as it was first described by Camillo Golgi. So this Golgi apparatus consists of a system of membrane-bound vesicles, which is flattened sacs. So they are arranged approximately parallel to each other as sacs called cystole. So these membranes often have connections with membranes of endoplasmic reticulum, therefore constitute another portion. So the material synthesized near endoplasmic reticulum is packaged and dispatched to various targets inside and outside the cell through Golgi apparatus. So it's the Golgi apparatus who is the carrier. Its functions include storage, modification and packaging of products. In some cases, complex sugars also may be made from simple sugars in Golgi apparatus. So Golgi apparatus is also involved in the formation of lysosome. So what is a lysosome? Lysosome, as it is told, it is a suicidal bag which actually digests any kind of waste, or that is a waste disposal system. So this helps to keep the cell clean by digesting any foreign materials as well as worn out cell organelles. So foreign materials, if it is entering your body, such as bacteria, food as well as old organelles, they end up in lysosome, which breaks the complex substance into simpler ones, and these lysosomes are able to do this because it contains powerful digestive enzymes which is capable of breaking down all organic material. So during the disturbance in cellular metabolism, for example, when the cell gets damaged, lysosome may burst and the enzymes digest their own cell. So that is why it is known as suicidal bag. Next we have mitochondria, which is called as the powerhouse of the cell. So it has ATPs. So they generate energy by chemical reaction. The energy required for various chemical activities needed for the life is released by mitochondria in the form of ATP, adenosine triphosphate. So mitochondria are strange organelles in the sense that they have their own DNA and ribosomes. Next we have plastids. We have basically the plastids are chromoplast and leucoplast and chloroplast. So chromoplast we have coloring pigments, and the green color pigmented ones is known as the chloroplast, which helps in the photosynthesis. We have leucoplast, which primarily are organelles in which materials such as starch, oils, protein granules are stored. Okay. Next we have vacuoles. So vacuoles are storage sacs for solids or liquids, for fats, carbohydrates, foods and all those. So in a plant 50 to 90% of the cell volume is vacuole. So the plant cell vacuoles are full or cell sap and provide turgidity and rigidity to the cell. Many substances of importance in life of the plant cell are stored in vacuoles. These include amino acids, sugars, various organic acids and some proteins. In single cell organisms like amoeba, the food vacuole contains the food items that the amoeba has consumed. In some unicellular organisms, specialized vacuoles also play important role in expelling excess water and some waste from the cell. So each cell thus acquires its structure and the ability of function because the organization of its membranes and organelles is specific ways. The cell thus is a basic structural organization, and this helps the cell to perform the functions like respirations and everything. So that is why the cell is the structural and functional unit of life. Okay. So then in the next video we will talk about cell division.